Hi there. In this video, we're going to walk through an example proteomics analysis on Pluto. Here we have a type 2 diabetes data set. So we actually have the proteomics and RNAseq data from a study on type 2 diabetes, where we've collected data from control, pre-type 2 diabetic, and diabetic patients. And what you're seeing here is basically the end result of that analysis. In this video, we'll really do a deep dive into the proteomics analysis, but I also want to talk about how you would go about integrating proteomics data with RNA-seq data to answer some of those translational questions. And then we'll also talk a bit about how to extract biomarkers from that data and manage biomarkers within Pluto. So what you're seeing here is the end result of this analysis, but you can see that the analysis started by just pulling in the counts data into Pluto. So I have my counts data as well as the sample metadata. And then from there, you can generate all sorts of analyses using non-coding graphical modules. So these are things like differential expression, gene set enrichment, and PCA. Once you have differential expression from two data sources like proteomics and RNA-seq, you can also generate concordance analysis, which is what we've done here to identify genes that were consistently differentially expressed across RNA-seq and proteomics data. So that's just a little high-level overview. This workspace that we're in right now is the Pluto canvas, and this is really where you'll spend the majority of your time analyzing data in Pluto, and it's an interactive space where you and your colleagues can work together on analysis. Everything updates in real time, really similar to a Google Doc or a Miro board. And then you can also write notes of your observations and leave comments on plots. So that's just a high-level overview, and now let's dive into the proteomics analysis. Okay, so within this proteomics experiment, the first thing you'll see is a notebook where you can record the objective and overview of your experiment. You can record the assay conditions, any notes, and planned analyses. This is a public data set, so I've gone ahead and included some information about where the data was sourced from. Down below, you'll see the sample metadata as well as the assay data. And for proteomics, that is really all you need to begin. You just need counts data, and that can be sourced from multiple different proteomics types like Olink or SomaLogic, for example. And when we have that data, we can launch our analysis. So I'll select this canvas here. You can see that I've already gone ahead and kind of created an example flow of an analysis here, but let's walk through how I did that. So usually the first question that you have when you start an analysis like this is, are my samples meaningfully different from one another? And to answer that question, we can create a PCA plot in Pluto. So I'm going to select this PCA and create analysis. And here you're going to want to compare all of the samples, but you could subset to just specific groups. So let's run that first analysis, and then it'll return this nice PCA plot to us, which we can go in and kind of adjust. So I'm going to go ahead and change this to be colored based on computed disease. And let's change the control to be blue, pre-type 2 diabetes to be kind of an orange color, and type 2 to be red. And then we'll see that there actually are some patterns appearing. So we have this control group kind of running through this circle here. And then the type 2 diabetes group is in this, we could lasso it kind of here. And then the pre-type 2 diabetes sort of falls in the middle. If all of these points were clustered into one kind of like tight circle, we probably wouldn't reveal anything interesting by doing subsequent analysis, but we do see that there's some heterogeneity in this data here, which means we'll probably find something interesting. So once we've done a PCA, typically the next step after that is to actually start looking at changes in gene expression patterns between our different conditions. And to explore that question, we want to run differential expression. So I'm going to select differential expression. And we are probably interested in looking, or at least I'm really interested in looking at if there is a difference between the type 2 diabetes and pre-type 2 diabetes condition. We may want to look at biomarkers of those specific stages of disease. And we may also want to identify, what gene pathways are changing from pre-type 2 to type 2 diabetic conditions. And so the starting point of that analysis is really differential expression. Again, I'm going to just go in and change some colors, and I may also want to change the adjusted p-value. So let's say I'll take that down to 0.05, and that'll reveal more significantly differentially expressed genes, or proteins in this case, and then change the colors. So type 2 is supposed to be red, and pre-type 2 should be orange. We may also want to examine specific sets of genes that we are interested in. So for example, I have followed this KEGG type 1 diabetes list, so I can save that, and then all of those genes will get labeled at the same time. And I can see, which of those are upregulated or downregulated between the conditions. So let's connect our PCA to this plot here. And then the next question I might want to ask is, are there actual pathways, driving these changes between pre-type 2 and type 2 diabetes. And because I've run a differential expression, from there I can run a gene set enrichment, which actually requires differential expression as a prerequisite. So I'm going to select gene set enrichment, and then I'll select the comparison that I just created, which was type 2 versus pre-type 2 diabetes, and then I'll select this hallmarks collection. So what this is going to do is run through every single gene set in the hallmarks collection and identify if those genes were, demonstrated enrichment in this comparison of type 2 versus pre-type 2 diabetes. And it will return the most enriched gene set, which in this case is hallmark interferon gamma response, which was more enriched or more highly expressed in the type 2 diabetes condition relative to pre-type 2 diabetes. But it'll also show us all of the gene sets that were identified as enriched. So an interesting one might be this hallmark pancreatic beta cell collection. So I could update and view that one. I could also view this as a waterfall plot, so I could look at all enriched gene sets simultaneously. So let's do that. And then I can see all of the gene sets that are positively enriched in the type 2 diabetes relative to the pre-type 2 diabetes. I'll go ahead and change that color as well to be red. In addition to running differential expression, you may also want to overlap specific comparisons. So for example, I have here taken pre-type 2 diabetes versus control and type 2 diabetes versus control, and then compared those via a Venn diagram. So because there were no significantly differentially expressed genes in pre-type 2 diabetes relative to control, I actually just lassoed a bunch of those genes and created my own list, but then I compared them to the differentially expressed genes in type 2 diabetes relative to the control. So then you can see genes that are just expressed or just significant in the pre-type 2 diabetes condition, or just significant in the type 2 diabetes condition, or shared across both, to help you kind of get a snapshot of each of those disease states. And then finally, you may also be interested in looking at absolute expression changes rather than log-fold change expression changes. So you could use a bar plot, a box plot, or heat map to look at the actual counts data as opposed to fold change data, which you're looking at with differential expression. Okay, so that is kind of a flow of proteomics. Now let's talk about how we could go about integrating that data with RNAseq. So I'm going to return back to my multi-omics canvas. And in this experiment, I have just linked my proteomics data set that we were just looking at and my RNAseq data set from the same study. And I've generated the same differential expression plots across both. So I have my type 2 diabetes versus pre-type 2 diabetes from my proteomics data set, and then the exact same comparison in my RNAseq data set. And then if I want to run multi-omics analysis on this data, I can click on Create New Analysis, select Multi-omics, and then select Concordance Analysis. So I can go in and choose my type 2 versus pre-type 2 diabetes proteomics comparison. So this is the actual differential expression from the proteomics data set of type 2 versus pre-type 2 diabetes. And then I'll select that exact same comparison from my RNAseq data set. And then that analysis is going to reveal which genes were significant in both comparisons, which were significant in only one comparison, and which were significant in the other, and then which genes were not significant at all. And the genes that we're really interested in here are the ones that fall along, well, typically it's the ones that fall along the line of equality that show us, genes that are shared across data sets. So this is really interesting to us if we're asking the question, is the RNA expression a good proxy for the protein expression? But we may also be interested in, seeing, are there any genes that are demonstrating that have post-translational modifications? And so the RNA and the protein are not concordant in that case. So depending on which question you're asking, there are different kind of groupings of genes that may be interesting to you. When you do identify genes that are relevant to your biological question, you can label them on this plot just by clicking on them. You could also search them here in this menu. And then when you want to save that as evidence, you can save these to a list. So let's select a biomarker list and select RNA and proteomics target selection results. And then we may also want to save this as evidence, or save this specific plot as evidence for one of those or multiple of those markers. So I'm going to X out of this, and then I'm going to save that plot as evidence to my new list. Okay, so I'm going to select my list that I just created, and I'm going to save it as evidence for this specific biomarker. And then when I go back to that biomarker in my databases, I can select that biomarker and see that that plot has been saved as evidence. So just to review, in this video, we covered how to analyze proteomics data in Pluto, how to integrate that data with RNA-seq data, and then how to identify biomarkers and store and manage those in Pluto as well. I hope this was helpful, and I will see you in the next video. Bye.